Welcome to today's episode on Neuron Café.
Welcome to This Explainer. Today, we're diving into a fascinating anthology of recent breakthroughs that reveal how our nervous system develops, how our behaviors are wired, and how the scientific process itself actually self-corrects. Honestly, we're going to treat these hard science findings almost like an unfolding narrative. Think of it like a beautifully animated Studio Ghibli film, but on a microscopic scale, exploring the living, breathing biology that makes us who we are, with all the wonder and awe of a grand story. Here is today's agenda. We've got 1. The dynamic nervous system. 2. Wiring the sense of touch. 3. Correcting the microglia record. 4. The neurobiology of overeating. And 5. An evolving science. Let's just jump right in. Section 1. Wiring the sense of touch. The mystery of sensation. We take our physical sensation completely for granted most of the time, don't we? But a massive, massive question in neurobiology has always been how exactly these highly specialized sensory pathways actually form early in life. I mean, think about how chaotic the developmental environment is. You have billions of cells migrating, dividing, trying to find their exact proper targets. Amidst all that biological noise, how does a developing nervous system actually know how to wire the precise mechanosensory end organs that let us feel a gentle breeze or a soft texture? It's honestly borderline miraculous. Well, it turns out the answer heavily involves something called piezo-2. This is the principal mechanosensitive ion channel in our somatosensory neurons. You can think of piezo-2 as a microscopic mechanical sensor. When physical force is applied to a cell, this little channel pops open to let ions flow. But recent findings show that piezo-2 isn't just sitting there waiting to send touch signals later in life. It's actually absolutely required for the structural maturation of our touch receptors during development. And this comparison brilliantly illustrates the cause and effect. Just look at the beautiful, highly organized green trails of neurofilaments in the normal sensory structures, the Meissner corpuscles, in the control mice. Then contrast that with the piezo-2 mutants. When you remove this mechanical sensor, those mechanosensory end organs completely fail to wire properly. They're disrupted, they're disorganized, and they are functionally impaired. So here's the absolute game changer. Physical activity itself shapes sensory development. The simple act of mechanically evoked neuronal activity early in life literally wires our ability to feel. The researchers proved this by deleting another electrical pathway, the voltage-gated sodium channel Nav1.6. By removing this channel, which normally propagates electrical signals, they disrupted these structures too. That confirms that the electrical firing triggered by mechanical stimulation is what actively guides the anatomy. Plus, when they used single-cell RNA sequencing to peek into the genetic code, they found that the absence of piezo-2 actually alters gene expression in these sensory neurons, meaning mechanical force isn't just pushing cells around, it's altering their genetic destiny, turning specific developmental genes on and off to literally write the physical architecture of our nervous system. Okay, moving on to Section 2, correcting the microglia record, the self-correcting process. Just as our nervous system adapts and develops, so does the field of neuroscience itself. A few years back, a really celebrated paper made this bold claim that introducing a factor called NeuroD1 could magically reprogram microglial cells, which are the vital immune cells of the brain, into fully functional neurons. As you can imagine, it was hailed as a huge potential breakthrough for brain repair. But a meticulous re-examination by a team of researchers found this to be entirely false. The corrected finding reveals that NeuroD1 cannot induce this conversion. Instead, it actually just causes the microglial cells to die. In science, replication and visualization are everything, right? If you look at these day 14 cell cultures, it's pretty clear. If reprogramming had actually occurred, we'd see a thriving population of mature neurons right here. But the ectopic expression of NeuroD1 clearly does not result in newly formed neurons. The specific markers you'd need to identify healthy, reprogrammed neurons, they just aren't there. The magic trick was unfortunately just an illusion. So instead of a magical cellular reprogramming, we get Exhibit A in the correction. By tracking the cells over 120 hours, the researchers proved that the manipulated microglial cells simply experienced apoptosis, which is just programmed cell death. You can see the cell count drop drastically compared to the control group. It's a sobering reminder that biology is profoundly complex, but honestly, it's also a beautiful testament to the scientific method. When errors happen, rigorous peer review and follow-up studies correct the record, making sure our understanding of the brain stays robust and accurate. All right, on to Section 3, the neurobiology of overeating, the battle of the brain.
To really understand overeating, we've got to start with basic brain metabolism. Our brains are incredibly energy-hungry organs. Think of the brain as a high-performance engine that's just constantly running at the red line. It desperately demands constant fuel, using glucose as its primary energy source, to strictly maintain our energy homeostasis. This exchange of metabolites and lipids between the brain and the circulatory system is a tightly regulated survival mechanism. Put simply, your brain is hardwired by millions of years of evolution to make absolutely sure you consume enough calories to stay alive.
But here's where the plot twists. Our ancient biological wiring is currently colliding head-on with the modern world. This quote really sums it up perfectly. Palatable food of high caloric density may override metabolic needs and cause a loss of control in regulating food intake. When we're consistently exposed to a modern environment that's just filled with highly palatable, hyper-processed, calorie-dense foods, our basic metabolic homeostatic needs get entirely hijacked. So let's explore how this builds a complete picture of modern eating behavior. This hijacking creates a profound neurobiological conflict between homeostatic feeding, which is eating purely for survival and energy balance, and hedonic feeding, which is reward-driven consumption that overrides that homeostasis. The drive for hedonic feeding can become so overwhelmingly strong that it actually parallels drug addiction within the brain's reward system. It shows us that overeating isn't just a simple lack of willpower, it's a complex, maladaptive neural behavior. And this is exactly why new pharmacological therapies, like GLP-1 receptor agonists, are proving so incredibly effective. They essentially step in as chemical circuit breakers, mimicking natural satiety hormones to quiet down all that reward-seeking noise. Similarly, emerging treatments using NMDA receptor antagonism work to block specific excitatory pathways in the brain, helping to reset the neural circuits that our modern food environment has completely knocked out of balance.
Which brings us to Section 4, in Evolving Science, our conclusion. So, what have we actually learned today? Well, from the delicate, activity-dependent wiring of a mouse's paw to the rigorous self-correction of scientific literature on microglial cells, all the way up to the complex, competing neural circuits that govern human food addiction, neuroscience is a vibrant, constantly evolving narrative of discovery.
I want to leave you all with a bit of a provocative thought as we wrap up this explainer. If understanding the deep neurobiology of overeating proves that our brains are easily bypassed and essentially hacked by modern diets, how should that change the way we live? If it's a matter of deeply ingrained biology rather than just willpower, should society change how it regulates the availability and marketing of these highly palatable foods? It's a massive question, and it sits right at the intersection of neuroscience, public health, and our everyday lives. Thank you so much for joining me on this journey through the nervous system, and remember to keep questioning the science around you.